Alright, so, title of my talk is Skeletal Muscle Epigenetics, Current Understanding and Future Directions. So very briefly about me, I'll just breeze through this. I've been here once before, I've done this lecture, that was the second one to do it, so if you really want to know more about me, you can go back and look there. But I got my undergraduate degree at UNC Chapel Hill, my master's degree in exercise physiology at James Madison. Then I got my PhD at Ball State University, where I did a project that was a collaboration with NASA, where we were essentially trying to figure out the best exercise prescription for astronauts, and Jimmy was involved in that as well, Dr. Bagley. And now I'm a postdoctoral fellow at the University of Kentucky. I've been there for about 5 years, and hopefully I will be transitioning out within the next couple of years to start my own lab. So that's that. The title of my talk is Epigenetics, so maybe we should get on the same page about what epigenetics actually are. Some definitions are in order. So broadly, the epigenetics is the study of changes in organisms caused by modification of gene expression rather than alteration of the genetic code itself. So this can happen in a couple different flavors. Two I'm going to focus, or one I'm not really going to focus on more than the other, but the first one is chromatin remodeling. So DNA is wound around these chromatin that can be modified in a variety of different ways, acetylated, methylated. All different types of modifications can happen to the chromatin, which essentially can help to dictate what genes actually get expressed. So depending on how the chromatin is being modified, transcription factors can be more accessible or less accessible to the DNA, and this can increase or decrease gene expression. Very complicated thing, but that is one flavor of epigenetic modification. Another one is DNA methylation, which is the one I'm going to focus on a little bit later in the talk, so keep this in mind. But DNA methylation is essentially the attachment of a methyl group to a cytosine in the DNA. And what that does is when a gene becomes methylated, it can modify how much it's expressed. Specifically in the promoter regions of genes, they can be methylated in a way that really has a very profound effect on gene expression. So as an example, if a specific gene is hypermethylated, so has a lot of methylation in the promoter region, it will repress how much that gene gets expressed. So the message, the mRNA at that level, will be lower. However, if a gene is hypomethylated, has less methylation in its promoter region, gene expression will go up. It means the gene is more accessible to transcription, and that gene can actually go higher in the cell. And so that's kind of how that works. So hypermethylation will repress gene expression of the promoter region, and hypomethylation will have the opposite effect. So I'll come and revisit that a little bit later in the talk. But I'm going to talk about skeletal muscle, and I think it's really unique and interesting. And actually, this is a nice photo that Dr. Bagley provided for me. It shows a muscle fiber, and a muscle fiber is this long cylindrical tube, and within it are many, many nuclei, or myonuclei, muscle nuclei. So I'm going to say myonuclei, and that means a muscle nucleus, or a nucleus specifically in a muscle fiber. This is really unique because most other cells in the body do not have multiple nuclei. Most cells in the body have one nucleus. So skeletal muscle is super unique because it can be peppered with myonuclei, have hundreds, perhaps even thousands, depending on how big the muscle cell is. So that's really unique. Another thing that's interesting is that these nuclei inside the muscle, the myonuclei, are post-mitotic, which means that they can't divide and replenish themselves. But they can be gained and lost through a couple of different mechanisms, which I'll touch upon briefly. But essentially, if you gain or lose more nuclei, you're changing the amount of DNA in the cell, which is really unique because other cells can't do that because they only have one nucleus. Muscle fiber has many nucleus. You can gain nuclei, you can lose nuclei, so you can change the overall DNA dosage within a muscle cell, which is a very unique feature of muscle. And this could theoretically be considered an epigenetic modification. If you add or subtract myonuclei, you change the amount of DNA, and that could conceivably change gene expression without changing the DNA code itself. So this is one unique theoretical epigenetic modification that could happen in skeletal muscle, and only in skeletal muscle. So we're all on the same page with epigenetics now, right? Later on I'll talk about methylation, hyper and hypo of the promoter, and right now I'm going to focus on the number of myonuclei and how modifying that could be a feature of epigenetic modification. So, there's this idea. Let's say that you want to go to the gym, you're going to start lifting weights. This is pretty much, for the most part, what would happen. So this is a cartoon of a muscle fiber. As you can see, it's a long cylinder, has multiple nuclei in it, it's multinucleated. What would happen is you'd start training, and the number of nuclei in your muscle would increase. How this happens is through something called a satellite cell. This is a stem cell that resides in muscle. As I stated previously, myonuclei can't replenish themselves. They can't divide. One myonucleus can't become two. So what you have to do if you want to increase the number of nuclei in a skeletal muscle fiber, you have to fuse in a new one from a satellite cell. So these muscle stem cells can be donors of myonuclei. So as you lift weights, as your muscle starts to get bigger, these stem cells will become activated and start fusing more nuclei into the muscle fiber as the muscle grows. The idea being that you need to be able to maintain the correct amount of gene product, the correct amount of mRNA, transcription, so to say, within the muscle fiber as it grows. And if you don't have enough nuclei to manage the transcription that's required for growth, then you might not grow as much. So that's at least the theory. So you start lifting, muscle fiber, you add more nuclei, muscle fiber gets bigger, and ultimately, once you're well trained, you have a bigger muscle fiber that has a lot more nuclei in it. See? So let's say something happened. You get injured, or you don't want to train anymore, you become lazy, you go on vacation, whatever. What would happen is that the muscle fiber would get smaller, but there's been some data, some evidence to suggest from different lab groups that you don't lose the nuclei, that the nuclei stay in the muscle fiber, so that the muscle fiber becomes quote-unquote hypernucleated. So muscle fiber gets smaller, you're detraining, you end up with a smaller muscle fiber that has more nuclei in it. Let's say you want to start training again. You get motivated again, you're not injured anymore, whatever the case may be. You start training again. The idea would be that you don't have to go through this process anymore, fusing in myonuclei, adding them in, putting them in the right place, whatever. You can just go right back to hypertrophy, go right back to getting larger. And there's a variety of different lines of evidence that have shown that after you've already trained and then you detrain, you can grow faster after that initial training period. And this is called quote-unquote muscle memory.
Is there a time or is there an expiration date on that hypernucleation?
I'm getting that.
Perfect.
and so that's the idea, though, is that there's this muscle memory that's related to the number of bionuclei. You don't lose them, so you can just grow really readily again. And I think anecdotally, a lot of people have noticed that once you stop training and train again, that it's easier to gain muscle back. I think a lot of people would probably agree with that, and there is some literature to support that idea as well. So where did this idea originate, and how strong is the evidence? I'm a big fan of trying to understand where ideas come from, and a lot of times you can look in the primary literature and figure that out. This was among the first studies to kind of address this phenomenon and put this idea out there that this is how quote-unquote muscle memory works, and this is some epigenetic mechanism of how the muscle remembers that you were exercising previously and then allows you to adapt more readily. So this is a study done by the Christian Gunderson lab group over in Norway. It was done in 2010, published in PNAS. And what they did to test this was they used a mouse model. And the problem with using mice model with exercise studies is generally that you can't really exercise them. They don't like to exercise. They're not like motivated in that way. You can't really train a mouse to do things, to pick up a dumbbell. It just won't do it. And they're very stubborn. And so in order to get a muscle to grow, if you want to study muscle hypertrophy in mice, you have to typically use a surgical technique. The technique we use is called the Synergist Ablation. And this is the technique they used as well. Essentially what happens is this is meant to represent the lower hind limb, so the calf essentially of a mouse. Essentially what people go in and do is they'll go and cut out a large portion of a certain muscle. In this case, it's the plantar flexor, so the muscle that's responsible for doing this. And so you'll cut out a portion of this muscle, but you'll leave one muscle behind that has a similar function, so a synergist muscle. That's why it's called synergist ablation. What happens is if you cut out these big muscles, you leave behind a smaller one. The smaller one has to compensate. So every time the mouse takes a step, it's not using the big muscles anymore, it's using the small one. So the small one has to become big in order to compensate for the mouse to continue to function. So this is called synergist ablation. So this is what they did. They did synergist ablation, they got the mouse to quote-unquote resistance train and grow, and then in order to get the muscle to quote-unquote detrain, so they can look at this muscle memory, see if the nuclei were permanent, they went in and they denervated the muscle. So they opened the muscle back up again, or opened the mouse back up again, and surgically resected part of the nerve that innervates this muscle. So then it atrophies, because it's no longer getting signal to contract. So it's a pretty clever little model. Synergist ablation followed by atrophy. So this is what they found. On the y-axis here is cross-sectional area. This is the size of the muscle fibers. On this y-axis here is the number of nuclei per muscle fiber. And if you recall, usually as the muscle grows, nuclei get added in from satellite cells. So here's what they found. That up here, or this line here, the solid line, is fiber CSA. So it's the size of the muscle fibers. It got bigger during the synergist ablation, the training period. But then once they detrained them, or once they cut the nerve, the muscle fibers got smaller, which would be expected. Hypertrophy followed by atrophy. They also found, though, that the number of nuclei increased during the hypertrophy, so during the training phase, but then during the detraining phase, the number of nuclei didn't go back down to baseline. It stayed relatively constant. So what they concluded here was that the nuclei you gain are then permanent, and they represent the muscle memory of previous exercise exposure. So the trick is with this, though, once you denervate the muscle, you can't go re-innervate it and then get it to train again, so that was kind of a bummer. So how do we resolve this issue in the context of exercise? We were really interested in exercise, right? That's my whole background is exercise. And it's, as I stated previously, we really wanted to be able to exercise mice, but typically to study muscle hypertrophy in a loading context, an exercise context, we have to use the surgical technique. So we instead developed this new technique, and this is in my view, one of the first true exercise models for mice. We called it progressive weighted wheel running, and this method was developed in collaboration with my postdoctoral fellow colleague, Dr. Corey Dungan. And we ended up publishing this paper already, and I'll show you some of the data, but it ended up working out really nicely. Essentially what we do is we take these mice, we put them in a wheel, they'll run voluntarily 8 to 10 kilometers a night. So they'll run a lot if you give them the opportunity to. What we ended up doing was we ended up weighting the wheel just on one side. And so typically people have used weighted wheel running before to exercise mice, but the problem is that once you get to a certain weight, they'll just stop running. It becomes too hard for them to even get the wheel started, and they just bail, and so they won't run at all. And so they stop exercising after a certain point, and then it's hard because you need to continue weighting them like progressive overload, but if they just stop running after a couple grams, then it's useless. And so we weighted the wheel just on one side by accident to begin with. Ended up working out really, really well, so we stuck with it. And so it utilizes an unbalanced wheel. We're able to gradually increase the weight over time. So a mouse weighs about 30 grams. We'll start with 2 grams and get them all the way up to 6 by the end of training. It's minimally injurious. With the Synergist Ablation, obviously we're cutting them open, we're cutting out muscle, we're doing all these things, it's damaging the muscle. We pretty much eliminate that. It trains the whole hind limb, which is great, so we can look at more than just one muscle. Before we could only look at one muscle, now we can look at the soleus, the plantaris, the gastroc, all these different muscles, which is great, so we can look at different fiber types. It encourages continued training. They'll run 10 kilometers a night, even with like 20 or 30% of their body weight on the wheel, which is amazing. And the adaptations are reversible, perhaps most importantly. We just lock the wheel, now they can start detraining. So it works really well. And I have this video here, maybe it'll work. That's always a mixed bag. Yeah, here we go. Okay, so you see they start running, the wheel is unbalanced, so they'll run, and then they'll get caught up. Whoop, around the wheel. Okay, it's like, okay, now I gotta stop. Okay, now I'm gonna start over then. Keeps going, run, whoop, around the wheel again. Okay, stop. They'll do that all night. They don't always flip over like that, but this guy was really feisty. But what'll happen is they'll just do that all night. They're like I said, they're not the brightest creatures, and so they'll just stop and start, almost like they're doing sets and reps all night long. So it's really great, and it ended up working out very well for our study. So now we have an exercise model. We developed this, it works. So, and I'm going to show you the data of it working. But we wanted to answer this question about training, detraining. Are myonuclei permanent, and does this represent some type of muscle memory mechanism? We can actually get at this now. So, first thing we did was we looked at muscle fiber size. We wanted to make sure they grew. And so we looked at the soleus, which is an oxidative muscle. It has type 1 and type 2A fibers. It's more oxidative. It's more tonically activated or chronically activated, tonically activated. So it's a muscle that perhaps a little more translatable to humans because we have type 1 and 2A fibers as well. So that's good. So we end up looking at soleus muscle fiber size. These are immunohistochemistry cross sections where we do the fiber typing. So we cut the entire muscle and we can stain it and look at the different fiber types. And these particular images here, the pink fibers are type 1 fibers and the green ones are type 2. What happened with the power training, muscle got much larger and they picked up way more type 1 fibers. So there's a fiber type shift towards more oxidative. And that's what happens with training. That's a trained muscle phenotype, larger, more oxidative. And then we detrain them, lock the wheel, and then we saw, that's weird, they didn't detrain. The muscle stayed pretty much the same size and the fiber type didn't shift back. So that was somewhat problematic for our study. We think the reason why that happened, after 3 months of detraining, we locked them in the wheel for 3 months after they trained for 8. We think that what happened was they kept up their eating habits. We actually have the data to show this too. They kept up their eating habits. So they were eating more while they were training, and then we cut the wheel, we stopped it, and then they just kept shoving food in their faces, and they blew up. They got really big in a short period of time. And we think what happened is they just, because the soleus is more of a tonically activated kind of postural muscle, it just kept getting loaded by the additional body weight that they put on so quickly, and so it didn't actually detrain. And we traced them out to 6 months too, which is the follow-up paper that we're going to publish here soon. They still didn't detrain by 6 months because they just kept getting bigger, and so we think that it's going to be tough to detrain the soleus. We'll have to do a pair feeding study where we control the amount of food that they eat in order to try and get back at that. So that's okay though. We were able to focus on the plantaris. So in this particular image here, these are the green fibers are the type 2A fibers, and the red ones are the type 2B fibers. And so, which are these are more glycolytic. The 2B and 2X fibers in the mouse are more glycolytic. So not quite as translatable. But what we found was that plantaris muscle got bigger, became more oxidative. Then we took the wheel away. We fixed it for 3 months, and then they went right back down to baseline levels. So they did train. They did train. Fiber size went up, went right back down. So at least in the plantaris muscle, we were able to get some idea of this training, detraining response. So the next question became, do they keep the myonuclei they gained during growth? Quickly, before I show you those data though, the other thing that's really nice about our model is there's systemic adaptations, right? They're running 8, 10, 12 kilometers a night. They have weight on the wheels, being progressively overloaded. So they actually adapt in their whole body. Their heart gets really big. They actually demonstrate an athlete's heart. They have a thicker left ventricle. They have heart rate go down. And basically, they become athletes. We have athlete mice now, which is great. So we have this kind of athlete model. We can modify it in different ways. We can add more weight, have them train longer. It's really great. So again, for I think probably the first time, we're able to really start doing exercise studies in mice as opposed to really just having them do unweighted wheel studies where this, essentially they're just going back to what they would be doing out in nature. Like this is actual like exercise for them in a lot of ways. So it's pretty cool. So anyway, I just want to throw that in there, a little plug for the power training model. And so we want to look at the number of myonuclei though. That was the big question. So just to remind you guys, this is what we were expecting to see. We were expecting to see that even with detraining, that they would keep the number of myonuclei that they gained during hypertrophy. And the soleus, because the muscle didn't detrain, they didn't really lose the myonuclei either. The muscle remained trained, so it stands to reason that they'd keep the nuclei they gained. Plantaris, to our surprise, gained myonuclei with training, lost them with detraining. So in a more translatable, kind of physiological model of training, it didn't seem like the nuclei were actually being retained, which was, to our surprise, and counter to our hypothesis. And we measured this in multiple different ways as well. So this epigenetic mechanism of muscle memory and hypertrophy, at least in our mouse model using this, it didn't seem to hold up, which was interesting. We wanted to see if there was maybe a fiber type specificity. As I mentioned, we traced them out to 6 months in hopes that the soleus would detrain. It didn't. So there are actually quite a few, there's a decent amount of 1, 2A and 1, 2A fibers in the gastrocnemius muscle, which is the bigger muscle on the calf back here, at least in the mouse it's the bigger muscle. And so we ended up just going in and via IHC looking at those fibers with training and detraining as well, and wanted to look at the number of myonuclei specifically in the oxidative fibers, the thought being that oxidative fibers have a higher synthetic rate and maybe they would be more prone to keeping the nuclei around so they can maintain transcription. We found that untrained mice to power trained mice, significant increase in the number of myonuclei, but then with detraining, went right back down to baseline as well. So we don't think that there's a fiber type specific component of this myonuclear muscle memory either. And this is prelim. We just finished collecting these data, and this will go into a second paper that we're working on. So we don't think there's like a fiber type specificity though. So granted, these fibers could be totally different than soleus type 1 and 2A oxidative fibers. I don't know, but at least we can infer from this that perhaps there's not a fiber type specific component to this phenomenon. So I've talked about mice, talked a lot about rodents. Rodents are great models, love using them. But what does the evidence for myonuclear muscle memory in humans suggest? So, this same group that originally proposed this hypothesis about the myonuclear muscle memory, Christian Gunderson, a bunch of other folks, decided to do a human training study. So they took people, basically what they did is they had people come into the lab, young untrained people, took a baseline muscle biopsy, so went in with a needle, grabbed a little sample of their thigh muscle out, and then trained them for 10 weeks, so did resistance training just on one leg, and then took another biopsy, so that was the trained state biopsy, and then they detrained them for 20 weeks, took a third biopsy from that leg in the detrained state, and then retrained that leg for a shorter period of time, with the idea being that they would be able to get the muscle to grow, that it would increase the number of myonuclei that would remain during this period. Then during this follow-up period of a second training, they'd grow more quickly, and they'd have those myonuclei would contribute to that. And then as their control condition, they'd use the other leg. So they only trained one leg for all of this part here, and then they used the opposite leg just for that second part of training, so they could compare directly. So it was actually a pretty elegant study design, and should have been able to answer their question with it. Here's the problem. So this is fiber size here on the y-axis, and this is the time course over here on the x. What they found is that the muscle fibers grew with the initial training, as to be expected. However, they didn't detrain. That was the problem. That was one of the problems. Fiber size didn't actually go down with the detraining, and so if the muscle didn't really detrain, they're not really able to answer their muscle memory question. I guess the fact that the muscle didn't detrain means that there was a memory of the hypertrophy because it never went away, but not really quite the question they were going after. And so what they also discovered was that they didn't actually increase the number of myonuclei with training either, which may come as a surprise to you all because I already kind of framed this as you add more nuclei for hypertrophy. There's a lot of evidence that suggests you don't actually need to do that to grow, and that's a different lecture for a different day. But still, so they weren't able to really test their hypothesis using this model, which is a bummer because this study took a long period of time, recruited a lot of people, a lot of work, big study. And so, unfortunately, they weren't able to really adjust their hypothesis. Fortunately, though, they put up all their raw data online. And so, we had a different question. We wanted to go and look at their data a little bit differently and see if maybe we could extract some information out of it that they didn't put in their paper. So we went in, we downloaded their data set, we did some analyses. Basically what we found was that there was a group of 7 subjects that did increase the number of myonuclei with training. Cool. So we pulled those guys out, and we looked to see, okay, of the ones that increased the number of myonuclei with training, what happened when they detrained? The answer, they lost the myonuclei. Using both the methods that they published, you see an increase in the number of myonuclei from these subjects and a subsequent decrease, which dovetails with the mouse data that I already showed you. So again, it may not be true necessarily that there's a myonuclear muscle memory per se. Not to say that if you gain myonuclei for a period of time, they wouldn't be useful if you were to train again, but it doesn't seem like, again, that they're permanent, that it would last forever, you know? So, and then very recently, another lab group independently confirmed this, doing a study in older people where they trained them and then detrained them for a year. I just highlighted this because you can see the number of nuclei in the type 2 fibers goes up with training, goes right back down with detraining. And then just like yesterday, another study essentially showed the same thing that just got published. And so now there's kind of this like growing body of evidence to suggest, okay, maybe the number of myonuclei isn't the reason why you can regrow more quickly after a period of detraining. So this is all kind of recent stuff, and I'm sure it's going to be more evidence coming out for different things and more things for us to look at. But this myonuclear muscle memory hypothesis maybe just needs to be revised a little bit, because if the nuclei aren't permanent, maybe there's just a window of time where they could be beneficial. So there was another study done by a different group, Adam Sharples, Claire Stewart, and Robert Seaborn. This really nice study where they wanted to look at this muscle memory phenomenon, but just in a different context. They wanted to look at DNA methylation, which is something I talked about previously. DNA methylation can modify gene expression in a very robust fashion, but it's generally thought that it can be a very stable modification as well. So if you modify DNA methylation, it can be maintained for a very long period of time. So they wanted to do a study very similar to the one I just showed you, where they did a training and a detraining and a retraining, but instead of looking at the number of myonuclei, they wanted to look at DNA methylation levels. So that's what they did. They brought people in, trained them, detrained them, retrained them, and did genome-wide methylation analysis on the muscle biopsy sample. So they grabbed biopsies from all these different time points, extracted the DNA, and then sent it out for something called DNA methylation analysis, where you can look at essentially a good portion of the whole genome and different methylation levels across genes within the genome. So really cool stuff. I'm not going to bog you down with all this data, but what's interesting is they found that there was a whole series of genes that after the initial training, the promoters just remained demethylated throughout the entire training. So they were originally more methylated, so that means they were more closed down, less accessible, less transcription. With after that initial training, they became less methylated, more opened up, more available for that gene to be expressed. And that memory stayed throughout the entire training time course, which really captured my imagination. This was done in 2018, so a pretty recent study. And they also found that with that initial DNA demethylation event that occurred with the original training period, that gene expression correspondingly for those genes went up, which is what you would expect. So you see this, this in vivo phenotype as a result of the fact that the DNA was being methylated differentially in the muscle. So that was really cool. So the idea is that maybe you can modify the methylation status of your DNA, and that could serve as a muscle memory so that if you were to retrain later after detraining, those genes are already demethylated, and you can just ramp them up, and that would somehow contribute to faster hypertrophy or greater muscle growth with that retraining. So a really interesting idea and concept. So here's the rub. With every study that's ever looked at anything about DNA methylation in muscle, it's been done essentially in a way where either you grab a biopsy sample or you grab a muscle sample, you grind it all up, and then you look at the methylation status. And that can tell you something, but here's the thing. Even if you were to isolate out individual muscle fibers, there would be contamination on those muscle fibers. If you look at muscle, there's a lot of nuclei within the muscle fibers, but there's a lot of nuclei that are outside the muscle fibers. There's muscle stem cells, there's macrophages, endothelial cells, there's fibrogenic cells. There's all these different cells that reside in muscle, as well as blood. There's all these different cells that reside in muscle. So even if you go in and try to tease out an individual muscle fiber and do DNA methylation on a muscle fiber, you're still going to get contamination from different cell populations. So this is the muscle fiber here. These are the nuclei within the muscle fiber. These are cells that adhere to the muscle fiber, so probably like fibrogenic cells, macrophages, things like that. I actually genetically labeled the muscle stem cells. We have a mouse where we can genetically label cells different ways in vivo fluorescently. And so I used one of these mice just so I could get a look to see if the satellite cells were sticking. They were. And then I labeled the muscle fiber with this actin stain that we use. And so, so that's kind of the problem is if you want to understand what's going on within a muscle fiber, it's very difficult to do because it's really hard to get these myonuclei out without contaminating them. So that's kind of like the hurdle we need to get over. How can we solve this problem so that we can start looking at muscle memory or just epigenetics in general within the muscle fiber specifically? So, developed a model. John McCarthy, Dr. John McCarthy, who I work with, he's been one of my mentors for the last 5 years. Brilliant guy. He developed the human skeletal actin reverse tetracycline transactivator TEDHON histone 2B green fluorescent protein mouse. Very fancy. John's a smart guy. So, we're just going to call it the HSA H2B mouse for short. Essentially what we can do here is we have this mouse that's genetically modified. We can dose it up with this drug that essentially activates this histone to become fluorescent and get incorporated into the nuclei. Just in the muscle though, only in the muscle fibers. So it's a really clever model where we just take our mice, we breed them, we give them the drug, turn on the fluorescence in the myonuclei, take the drug away, no more labeling, it stops. Then we can extract the nuclei and look only at the muscle fiber nuclei. So this is great. You solve the problem. Really clever model. And so what's really nice about this is this has already been published, and the label efficiency is super, super high. We get about 95% labeling efficiency, and it doesn't really seem to label anything else other than myonuclei too. Sometimes there's off-target effects with these genetic models. Ours seems very, very specific. So we saw opportunity here. Now we can start doing some different analyses that otherwise weren't possible. So I saw this, I said, okay, I'm going to use this. This is the model that I want to use. So what you can do with it is you can extract the nuclei in a relatively simple fashion. So I developed this workflow where I take muscle, that's from the HSA GFP mouse, nuclei are labeled, just make a little nuclear suspension out of it, take it over to something called the cell sorter, a FACS sorter. So essentially it's this device that uses fluorescence and shoots cells in different directions depending on what color they are. So it'll just shoot my nuclei into one tube and shoot everything else into another tube. Really great. So I do that, and I also label the nuclei with something called PI, which is just a general stain. It will stain all DNA. So my myonuclei are going to be GFP positive, PI positive, and anything that's not a myonucleus will be GFP negative, PI positive. So that's really nice. So I get my two populations. I get my myonuclei up here, GFP positive, PI positive, and I get all other nuclei, which are GFP negative, PI positive. So I can get these two different populations. I can parse them out. So then I can look at them on the scope. Oh yeah, they're all green. They all have the DNA stain. I have myonuclei. Everything's good. Then I can go over here. I can extract DNA out of both those populations if I want to. Take the DNA, and then I can get these large fragments of DNA that are either specifically from myonuclei or from all the other populations, and I can send them out for all different types of analyses. I can analyze the DNA any which way. And so it's really nice because now we can really start digging into what's going on epigenetically in the skeletal muscle. So, before I get too crazy here, I said before that DNA methylation is typically viewed as kind of a stable modification. However, there's been a variety of studies over the last decade or so that have suggested that acute stimulus with exercise and muscle causes the DNA methylation landscape to change. So this was the first study that was published in 2012 and published by Julian Zierast's group, and she is super legit. And I was able to convince her to be a collaborator and a mentor on my K99 project, which I'm working on now. And so that was really great. So now I have this person who published his first study who's an expert in this, who's going to help me to understand this stuff. So she published his first study showing that acute exercise remodels promoters remodels DNA methylation of promoters with exercise in muscle. So they did this in muscle biopsy samples from humans. Again, they pulled out muscle samples, ground them all up, a lot of different things now, but showed preliminarily that, yeah, the DNA methylome can change with acute exercise. A couple other studies have done this too, the one I already touched on before, and Dr. Bagley published a little paper on this as well. So interesting that when you take muscle after exercise, the methylation landscape can change. So that kind of, captured my imagination too. So there's this guy, his name is Ferdinand von Walden. He is a MD PhD at the Karolinska Institute, and he's visiting University of Kentucky right now. I didn't really know who he was, but we crossed paths because he was in the lab that I was working in all the time. Hey, what's up, man? We started talking. Turns out he wanted to get into some DNA methylation stuff, and he found out about the model we had. And I was like, cool, he's like, you want to collaborate? I was like, yeah, let's do that. So we did a little study. The first thing we wanted to do was just answer a simple question. We want to look at myonuclear epigenetics with hypertrophy. We decided to do a short-term overload. The reason why we didn't do the power model is just we didn't want to look at training, and we hadn't piloted our acute power yet or anything, and so we just wanted to do synergist ablation. We know that this induces hypertrophy. We know it's a very, very robust model. So at least for a first pass, we're like, we'll use synergist ablation. We'll make that plantaris muscle grow. We'll take the labeled myonuclei, we'll sort them, and we'll do something called reduced representation bisulfite sequencing, which is essentially a way to capture a really good picture of the genome and what's going on at different methylation sites across many, many, many sites over the genome. So the first thing that was interesting that we found, so again, we ground up the muscle, it had the myonuclei were labeled, we take it over to the FACS sorter so we can sort out our different populations. Sham mice, so mice that didn't get the surgery, that weren't overloaded, so our control condition, only about 50% or so of the muscle, or of the nuclei were actually myonuclei. If I had to guess, I would have said 75, 80, 90% of the nuclei are probably still myonuclei. Very low. I was very off. It was only about half the nuclei muscle are myonuclei. Again, emphasizing how important it is to try and parse out these different populations. So only about half the nuclei are myonuclei to begin with. Then after 3 days of overload, that percentage drops to 30%. So 70% of the nuclei in muscle after 3 days of overload were not inside muscle fibers, were not myonuclei. Essentially, with the overload, you get some inflammation, you get some infiltration of different populations, you also get proliferation of stem cells, different cell populations, fibroblasts. So essentially, this proportion shifts dramatically because you have all these other things outside the nuclei, or outside the muscle, that are coming in and proliferating, and the nuclei themselves don't divide. So pretty much after 3 days, you get this huge reduction in the percentage of myonuclei. Again, emphasizing how important it is if you want to look in the muscle. At this early time point, mostly what you're seeing is everything else, not myonuclei. So that was kind of interesting. So that was the first thing. So we went ahead and we did this RRBS technique to look at tens of thousands of sites across the genome to see if methylation was changing, specifically in the myonuclei with this acute hypertrophic stimulus. Over here, this is just a heat map showing general differences between the two groups, and it was very striking. As you can see, this is the sham group here, this is the overload group here, and red means hypermethylation, so less methylation, and blue means hypo, so hyper, more methylation, hypo, less methylation. What happened is with the overload, the myonuclei the DNA in the myonuclei became way less methylated. Essentially, the DNA became way more available for things to transcribe and for gene expression to go up, which makes sense. The muscle's trying to adapt, so you have this huge demethylation response so that transcription, the amount of message of those genes can start increasing. So, and then I just went and did some pathway analysis to kind of get an idea of the different processes that were being modulated by this methylation response. I just focused specifically on the promoters because we know if you hypermethylate a promoter, gene expression tends to go down. If you hypomethylate, expression tends to go up. So I just wanted to look at promoter hypomethylation, since pretty much what we saw was mostly less methylation with the overload. And so I did some pathway analysis, and it was pretty robust regulation of P10 and PIP3 and TP53 or P53, basically things that are related to mTOR signaling, which is the master regulator of muscle growth, as well as autophagy, which is a really important process for cell quality control. So we saw this, which neither of these are necessarily surprising, but the extent to which I saw this was very surprising, at least to me. And then also there was this demethylation event in the promoter region of MYC, which is one of the master regulators of ribosome biogenesis, we'll talk about a little bit. But essentially what we found, which isn't super surprising, but again the magnitude, was that the things that regulate growth are being highly regulated at the level of DNA methylation. So this seems really important in muscle, which is cool and kind of gives us a platform from which we can move forward. So I did another thing. So this is just the same figure I just showed you. This is the myonuclei and kind of the global methylation status. We also took those interstitial nuclei. So all the nuclei that weren't muscle fiber nuclei, we grabbed those out too because I wanted to see how are those being methylated. Is it totally different than myonuclei? Is it the same? Is there overlap? Like if I just take whole muscle samples and grind them up, is it totally fine because these will just track perfectly with these? I didn't know. The interstitial nuclei were essentially the exact opposite. They became more hypermethylated and all the genes were essentially totally different and how they're being methylated as well. So it just goes to show that the different populations, myonuclei, other nuclei in muscle, are kind of acting in different ways. So that was interesting as well. So this is a paper that we have out in review right now. So I mentioned ribosome biogenesis. And Ferdinand von Walden, who's here visiting, is very ribosome biogenesis oriented. John McCarthy says he runs a quote-unquote ribocentric lab. Very interested in ribosome biogenesis. This other guy in the lab, Andre Casagrande Figueiredo, Brazilian fella, he really loves ribosome biogenesis too. It's definitely a focus of the lab. It's something we like to think about a lot. We think that ribosome biogenesis is important for skeletal muscle hypertrophy. So what is ribosome biogenesis? Ribosomes are the things that make proteins in the cells. The idea is that, let's say you start going to the gym, you encounter a hypertrophic stimulus. What would happen is at first the ribosomes you have could kind of pick up the slack. They would increase their efficiency. They would start transcribing more genes. But then there would come a point at which you need to start building more ribosomes, building more protein factories so that you can keep up with the adaptation that needs to occur. This is ribosome biogenesis, the making of new ribosomes. The idea is that if you're not able to do this, or not do it effectively, you're not going to grow as much. And that if you're able to drive more ribosome biogenesis, you might actually be able to grow more. So this is really what Ferdinand was very interested in, is this idea of ribosome biogenesis, epigenetic regulation of it in the context of skeletal muscle hypertrophy. So he came to me and we started talking, and it's like, yeah, this is perfect. You're the expert in this, I'm doing this, let's get together, we can try to answer this question. Let's look at ribosome biogenesis and DNA methylation in myonuclei. Boom. So that's what we did. So, and those same mice that I showed you, where I showed you the global methylation patterns across the genome, we also looked specifically at the rDNA, the ribosomal DNA. Ribosomal DNA is a multi-copy gene. It means a lot of genes, you have two of them. Ribosomes, you can have 50, 100, 200, varies from person to person, it's multi-copy. But we still wanted to look at it because we wanted to see if we could just see changes in methylation, even though it's a multi-copy gene. We still thought it would be interesting to look at. So in those same nuclei that I already showed you, we looked at the ribosomal DNA and found that sham is in black, overload is in gray, and this is just the level of methylation across the entire rDNA gene. Essentially what we found is that there's a global hypomethylation. So there's less methylation. Theoretically, this could result in more transcription of ribosomes and increased ribosome biogenesis. However, we know that in the mouse, there's one specific site in the ribosomal gene that's really, really important. And if it gets methylated or demethylated, it can really result in a very strong suppression or activation of ribosome biogenesis. And at that specific site, nothing happened. So that tells us that things are happening across the rDNA, but it may not necessarily be regulated at the level of methylation from a ribosome biogenesis perspective, potentially. So I have this mouse data. He goes, hey, I have all this human data from acute exercise studies, and I looked at ribosomal DNA methylation. We can put them together. I was like, great, let's do that. So he did pretty much a very similar analysis using muscle biopsies from human samples. So he had people come into his lab. He had them aerobic exercise, and then he had them resistance exercise. and then tracked them out over time in an acute study and just did a little time course up to about 24 hours, taking biopsies, extracting the DNA, and then looking at the methylation of the rDNA. The thought was that hopefully we'll see something similar, and sure enough, in humans, at least using whole muscle samples, using the biopsy samples, it didn't really seem like there was a difference in the major DNA regulatory sites in humans as far as methylation goes. So that actually nice, and they dovetailed together. And this is an instance where what we were seeing within the myonuclei did reflect what was seeming to happen with all the other cells that were involved too. So that's also kind of cool. So we have this human study now, we're able to look at DNA methylation in some different ways, compare it back to the mouse. It seems like ribosome biogenesis, the creation of new ribosomes with hypertrophy, is being regulated in a different way that perhaps is not related necessarily to DNA methylation, which is interesting in and of itself too. So, what Ferdinand went one step further though, as I mentioned previously, ribosome copy number, the amount of ribosomal genes you have in your DNA can vary. And so he wanted to say, okay, well, maybe it's not being regulated at the methylation level, but maybe some people just have more ribosome genes, some people have less, and this would dictate how much ribosome biogenesis you see after exercise. And sure enough, that was pretty much exactly what he saw. If you had more ribosome genes, you saw a greater increase in the amount of ribosome biogenesis after resistance exercise. So that's kind of interesting too. So we think that maybe this could be some sort of explanation for why some people grow more with resistance training and some people don't. Some people just have more ribosomal genes and can access ribosome biogenesis more readily and can grow more, potentially. So that's something we're really interested in looking at now too. So this is a second study that, again, we're probably hopefully going to submit sometime this summer. So yeah, that's a very fruitful collaboration. So conclusions and future directions. So first, power is a murine model of exercise training that is more translatable to humans than other approaches. We can use this as a tool, very powerful, no pun intended. Myonuclei gained during training do not appear permanent. That came as a surprise to us. Our hypothesis that they would be permanent, but it seems like perhaps they're not. A myonuclear muscle memory of hypertrophy does not appear to be fiber type specific, again, contrary to what we would have hypothesized, but still interesting nonetheless. Other epigenetic changes within muscle could explain enhanced adaptability to exercise after initial training period. So, instead of looking at the number of myonuclei, maybe we need to move to looking more into epigenetics, looking at histone modifications, looking at DNA methylation, things not related perhaps to the number of myonuclei per se. To understand epigenetics of skeletal muscle, it is very helpful to isolate myonuclei. We have that cool model, other people can use it, we're going to publish hopefully on this soon. So that's been really informational for us. So getting really in there within the muscle fiber to see what's going on, I think is really going to inform the path forward when we start studying epigenetics more in skeletal muscle. The acute myonuclear methylation response to a hypertrophic stimulus relates to mTOR and autophagy, and this may underpin the eventual increase in muscle mass. So there might be this initial methylation response in myonuclei that ultimately results in all the pathways that got upregulated during growth. I think this makes sense. Future studies will use myonuclear isolations to understand epigenetic muscle memory with training and detraining, something we're working on right now. We're going to train mice, we're going to detrain them, we're going to look at all this stuff specifically in the myonuclei as well as the non-myonuclei. So I'm really excited, and that's something I'm going to be funded to do hopefully for the next couple of years here. So that's the end of my talk. Quickly, I need to thank some people though. My postdoc mentors, Dr. Charlotte Peterson, amazingly brilliant woman. Dr. John McCarthy and Esther Dupont also mentors me in some capacity as well. Great people at the University of Kentucky doing awesome research, and I've had super awesome experience with them, so I highly recommend checking them out if this is the type of research you're interested in doing. My funding, I was first funded by NIH F32, and now I'm funded by an NIH K99, so that's been really big for my success. And their funding as well, they've had buku grants over the years, so and that has been really instrumental to me getting a lot of this stuff off the ground. My K99 mentors now, Dr. Julian Zerath at Karolinska Institute and Dr. Yvonne von Nuke-Mittendorf at the University of Kentucky. Brilliant women, and they are very good with epigenetics, so I'm going to learn a lot from them. I'm very excited. And a variety of other collaborators. I need to call out Dr. Corey Dungan, my fellow postdoc as well, brilliant guy. He's doing awesome work, and he was really the pioneer of power training, so I need to shout him out too. And so, yeah, I think that's about it. And I just got married last year, and the best day ever of my lovely life, so I'm allowed to be here.